Thank you very much for introduction. This is a great honor to present after such a great scientist. Today, I think the camp already introduced me. But just in case, I'm Yuji Nakada from UAB, it's a research hub. So today I will talk about title is already he mentioned, but so today I'm going to talk about basically single nuclear RNA-seq analysis. And so we basically did pig, and we newly invented like a double injury surgical model. So we talk about this RNA-seq model. So I think, as you know already, the ischemic heart disease affect over the worldwide, and 50% of patients die within 5 years of diagnosis. And key factor is the adult mammary heart is unable to regenerate the heart after the injury. So the first cardiac injury was observed in zebrafish. Sorry, newt in 1971 by Dr. Jones and James Oberfeuer. In the 2002 zebrafish was reported by Dr. Kenneth Foss et al. So after that, how is neonatal? And 2011, Dr. Hisham Sadek group reported, and both apical resection and myocardial infarction, the neonatal mouse, after the 3 weeks later, it completely degenerated. And unlike zebrafish, this degenerative window is limited to the 7 days after birth. So, and lower vertebrate and the neonatal small animal can regenerate the heart, but how about large animals? So 2018, Dr. Janie Dunn's group and Dr. Stuart Cook's group first identified the same time that neonatal pig could regenerate the heart after myocardial injury and through proliferation of cardiomyocyte. And this regeneration window is also limited, like 2 days after birth. And we keep thinking about, is there any good way to elongate, like extend this degenerative window? And recently we found that apical resection at P1 neonatal pigs prolonged the cell cycle activity and promoted the myocardial regeneration after left ventricular injury at P27. So this double injury pig can recover cardiac structure and function completely. As you can see here, the left panel is myocardial infarction only at P28. And 4 weeks later, we harvested and see the section. And as you can see, the scar, visible scar left. In contrast, the double injury model and apically resected at P1 and followed by myocardial infarction at P28. And after 4 weeks later, the cardiac, the scar, we did not observe any visible scar, and the echocardiac function is almost normal, and we didn't see the significant difference to the normal and myocardial infarction. And we also check the proliferation marker. We did several marker check, and this is PHC. Positive cardiomyocyte nucleus, and as you can see here, the double injury group significantly increased the PH3 positive cell. And interestingly, apical, the border zone and the remote zone, we see the elevated the proliferation marker. And we also observed the same thing in a similar result of 10Ki67 and Aurora B kinase. So we observed the cardiac regeneration and the function recovery in a double injury pig heart. However, the molecular mechanism of the regenerative window extension is unknown. And therefore, we try to address this issue. We performed the single nuclear RNA-seq analysis, and from the different the injury group and the time points. So at the P1, we did, as I said, apically resected, and 28 days later, the second injury received, which in this case is myocardial infarction. And after that, heart was harvested at different time point, and the process the nuclear processed and collected the RNA. So total of about, I think, the 290,000 high-quality nuclei were captured from animal groups and the time points, and about 95,000 of which are cardiomyocyte nuclei, and which indicated the orange color. And the cardiomyocyte group was identified by the cardiomyocyte specific markers, so myocyte specific markers. And we also identified other different cell type like fibroblast and immune cell and smooth muscle cell, T cell, mesenchymal cell, and the neuronal cell and endothelial cell. So today just we focused on talking about the cardiomyocyte. And the gene expression profile revealed that regenerating double injury heart contained a different subpopulation of cardiomyocyte after the second injury. And Rubin clustering identified 6 cardiomyocyte subclusters, as you can see here. And so this is a distribution map, and so distribution analysis show that the CM2 and CM3 and CM6 clusters have mainly comprised the P56 and the P1 and embryo. So almost the 96.2% and the 95.7% and the 92.4% respectively. And in contrast, the CM1 cluster have comprised heterogeneous cardiomyocyte population, and which from like all other animal group and the time points, as well as a small number of P35 double injury, and a limited number of the P1 and the fetal group. So, and the CM5 and the CM4, interestingly, they came from mainly P35 double injury group, 97.8%, and also 68.6%. And we also checked the G2M score, the cell cycle analysis. And actually, CM3 is neonatal-like cluster, and CM4 is DIPH dominant cluster, and CM5 is PKM2 dominant clusters, and the CM6 is a fetal-like cluster. And we found that CM4 and CM6, CM3 to CM6, this 4 cluster is significantly increased the D2M score compared to the more adult heterogeneous CM1 group, and also CM2 is adult. And interestingly, we are done the ketone mapping held the PKM2 population is in the CM5 cluster was significantly highly expressed to this CM5. And recently, I think Leo Zhangji group reported PKM2 is very important for cardiomyocyte cell cycle and promote cardiac regeneration. So, and PKM2 may play an important role in this double injury model. And so, and next we just also check the immunohistochemistry, how is the PKM2 expression that's conformed to the distant myocardial infarction only. In the double injury model, elevated the PKM2 expression, both cardiomyocyte and non-cardiomyocyte. And the messenger RNA, also PKM2, is significantly increased the double injury group. And so PKM2 is a glucolytic enzyme. So we wondered how is glucolysis is elevated or not. And first, this is the G6PD, the messenger RNA, the qPCR, the result, and the glucose 6 phosphodehydrogenase was elevated, the double injury model, which kind of makes sense because the PKM2 elevated the glucose lytic pathway, and also we more detailed analysis the different CM clusters, and the CM5 PKM2 dominant cluster was strictly close to the CM6 like embryo-like cluster, and also the neonatal-like cluster. So this means I think the G6PD is also known as the one of the glucolytic enzymes, especially like a first the enzyme of the PPP pathway. And so because of this activation of PPP pathway, probably PPP pathway also generate a sub-branch pathway of glycolysis pathway, and this PPP pathway will generate the pentose, like hexose, like for like a source of like a DNA synthesis. And not only that, but also the PPP pathway generate NADPH for important factor for maintenance of cell, especially like work as a reductase. So, and so we're curious about the reactive oxygen species, how changed in the double injury model. And we observed that double injury model is significantly slowing down the reactive oxygen species detected by the dihydrolodamine 1, 2, 3, and it's indicated in red color. And so since reactive oxygen species and the DNA damage is highly related, so next we wanted to see how is DNA damage was changed in the double injury model. So here is indicated by the 8-hydroxyguanine, which base excision DNA damage marker. Interestingly, double injury, I think as we expected, double injury model significantly lower down the 8-hydroxyguanine expression. And so we also examine the cell cycle analysis by the sparse modeling, the RNA-seq data, and just focus phase and the DTM phase, which is most important to cell cycle phase. And both phases are embryo and the P1 neonatal is highly activated. And adult population like P56 was not. And most the right side, the myocardial infarction P56, this one was not much activated, but instead the double injury group was highly activated, the S phase and the D2M phase. And in contrast, this is kind of opposite, like maturity, the cardiomyocyte maturity examination, and indicated by adrenergic signaling and the Z disc and cardiac muscle contraction, the gene population. And as you can see, the fetal and the P1 neonatal population is lower than this, like a cardiomyocyte maturity marker. And instead, the adult population is higher, of course. And the myocardial infarction only, this population also high. And however, the regenerative cardiac injury population, like double injury, and is significantly the lower the contraction maturity gene expression profile. So this suggesting like apical resection at the P1 make cardiomyocyte immature. So here's a summary. We have established the double injury pig model, and the first time to show extending cardiomyocyte regenerative window in a large animal, and transient expression of CM5 cluster induced by apical resection might contribute to demuscularization and functional recovery, and through glycolysis phosphorylation and the DNA damage reduction following the second myocardial infarction. And apical resection perturbed the cardiomyocyte maturation, which likely contributed to recovery from the second myocardial injury. So I'd like to thank the Dr. Jamie Zhang's group and also collaborator at University of Minnesota group. And this picture was just took by the Monday. And so hopefully like everybody coming back like this smile. So thank you very much. Now I take few questions.